In light of the great expansion of the pharmaceutical industry over the past 30 years, many drugs have been developed for promoting human health. During this development, many prospective drugs have been found to be carcinogenic in 2-year rodent studies. For reasons that are not published, the regulatory agency have approved for human use many drugs which cause tumors in rodent studies. For example, cholesterol-lowering drugs are rodent carcinogen. Atorvastatin gives tumors in rats at sarcomas and tumors in mice at the liver. Another example is pravastatin that gives tumors at the liver and lung. Other example also simvastatin gives thyroid and liver tumor in rats, while in mice the target site is liver, lung, Hendrian glands. There are other examples also, carcinogenicity bioassay of some other popular pharmaceutical. For example, lansoprazole gives tumor in rats at the testis, stomach, while in mice also attack at testis and liver. Metoprolol also gives tumors in rats and mice. Regulatory agencies use rules, regulations and guidelines for approval of new drugs for doses given to rodents in 2-year studies, which have been many times above the normal human therapeutic doses. Rarely, the rodent carcinogenic dose is only 1 to 3 times the human therapeutic dose. Many rodent carcinogens are FDA-approved drugs, primarily prescription drugs. Some of the drugs are multiple organ carcinogens in rodent, and few studies included doses barely above the human therapeutic dose. The information on toxicity or carcinogenicity in rodent is often obtained only from the product insert rather than from published report.
For the procedure of rodent carcinogenicity test for species and strain selection, in most studies, inbred rodent strains are used in order to reduce experimental variables and to enhance the interpretability of the results. Background data can facilitate the interpretation of experimental results. Rodent liver is highly susceptible for the induction of tumors by non-genotoxic chemicals. These tumors are not always relevant to carcinogenic risk in human and mislead the use of rodent for carcinogenic risk estimation. The rat carcinogenicity study is widely accepted. The route of exposure in animals should be the same as the intended clinical route. The target organ for the clinical route should be adequately exposed to the test material. Pharmaceutical applied topically with poor systemic absorption may not need studies by the oral route to assess the carcinogenic effect to internal organ. However, in case of photocarcinogen, dermal application, generally in mice, may be needed. There is a general agreement about the exposure time of 18 to 24 months for mice and at least 24 months for rats in lifetime carcinogenesis bioassay. Both the time frames agree well for inducing tumors because of continuous administration of test chemical. Lastly is the dose selection. There are toxicity-based endpoints, pharmacokinetic endpoints, saturation of absorption, pharmacodynamic endpoints, maximum feasible dose and additional endpoints.
Mechanisms of carcinogenesis in rodents and humans. Chemicals mostly cause or induce tumors in animals and humans by several mechanisms, most commonly referred to as genotoxic and non-genotoxic. Genotoxic chemicals may cause tumors by directly or indirectly damaging DNA and a genetic change that may end in neoplasia. Genotoxic chemicals can also be toxic at high dose, causing tissue damage that may contribute to promotion of carcinogenesis initiated by DNA damage. Non-genotoxic carcinogens often give high toxic doses to rats and mice, causing chronic target organ lesion, which are suggested to be the cause of tumor found in this tissue after chemical exposure. Associated with the chronic toxicity is chronic regenerative lesion with enhanced cell proliferation and turnover. This led in increased rate of cell proliferation and other mechanisms involved in chronic disease that will promote carcinogenesis in rodents and humans.
So for the observation and result, we will observe the clinical sign and mortality and also observing the body weight and food consumption. This will be recorded weekly. Also the mean body weight of the animal will be compared between those group and also the control group. The increase in body weight will be linked to the increase in the tumor rate in the mice. For toxicokinetic study, blood sample will also be collected at various intervals in morning and also afternoon. At the end, all the animal will be undergoing the necropsy that include weighing and examination of several organ and tissue. This is to make sure whether there is a tumor in some of the organ, and the frequently recommended organ are liver, kidney, adrenal, uterus, and so on.
So there's a number of advantages. Since we use the maximum tolerated dose, which is most likely to induce tumors, it provides information about tumor type and about which organs are sensitive to the carcinogenic effects of the drug. The second advantage is when the bioassays are conducted in more than one animal species, it provides a consistent basis for interspecies comparisons, which enables us to extrapolate the data from the animal test to humans and also allow us to predict the results in humans too. Another advantage of maximum tolerated drug is that it provides information on a number of endpoints of toxicity in addition to cancer. We have some disadvantages too. First one is rodents generally are not designed specifically to provide information on biochemical and physiologic mechanisms operating during the production of tumors. The second one is that it is very expensive and time consuming. And the last disadvantage is that the failure to account for biologic mechanisms of action could lead to errors in qualitative and quantitative predictions about human carcinogenesis.